Sorry. Welcome to Dr. Sophia Hirakis.
I just requested to share my screen.
There we go. And I think, okay, sorry. Dr. Hirakis received her Bachelor of Science in Biochemistry at Manhattan College and excuse me, her Master of Science in Chemistry here at UC San Diego. And she also received her PhD with the interfaces specialization in multi-scale biology. It was a chemistry degree with the multi-scale biology specialization, also here at UC San Diego in 2015, working in the lab of Dr. Rami Amaro and Dr. Terry. Is that Sejnowski?
Zanowski.
Zanowski. I've heard it a couple of different ways and I've not met him yet. At the Salk Institute. So please join me in welcoming Dr. Hirakis.
Hi, everybody. First of all, thank you so much for the distinct honor of being able to present to you all today. I absolutely loved my time at UCSD and at the Interfaces graduate program. I was very, very fortunate to be part of it. And I wanted to tell you all a bit about how my work was directly influenced by the training program and hopefully influence you to leverage the strengths that you will gain as part of the program. So just a brief outline of what we're going to be discussing today. We're going to start, most of the presentation is going to be the first part about how the coursework and my work were directly linked. I have a PhD specialization in multi-scale biology, and I really leveraged the coursework in this program, and it directly influenced my work. And then in the second part, I'll tell you a little bit about how I developed professionally in the training program, specifically through being able to teach in some of the courses that I took, and also being able to influence the renewal of the training grant. By writing some of the renewal as well. And in present day, I work at the U.S. Patent and Trademark Office, and the Interfaces training program has most definitely influenced the way that I interact in the professional sector in the government. And so with that, we'll start with that very first part about how my coursework was specifically influencing my research. And as we all know, as you all very well know, biology is multi-scale. Things happen at different time and space scales. So many of you will have learned about the cardiac system, which is one of the systems that I worked on in my graduate career. And if we're thinking about the smallest space scale, you can get all the way down to the atomistic scale, which is the scale of angstroms. When you're thinking about small molecules, for instance, like this is showing CAMP in the specific slide. And if you're going further up into the nanometer scale, you're thinking about the size of proteins. But if you're going further up to see how cellular systems interact with each other, you have to really start to think about the micron scale. And then if we're getting to the scale of the heart, and we're talking about millimeters, and then of course, humans are on the scale of meters. So the goal of the Interfaces training program is to sort of unite all of those space scales and work at those interfaces to be able to integrate all that data. And with that, as you all very well know, the goal of the Interfaces program is to specifically train a new generation of scientists to think in cross-disciplinary ways. And the way that they do that is through formal collaborations through different departments at UCSD. I specifically am from the Department of Chemistry and Biochemistry, but I interface very closely with bioengineering, biological sciences, neurosciences, and physics, both with professors as well as with colleagues. So it was such an enriching experience. So there were a number of courses that I took, and there are a number of courses that are offered to you today, but today specifically, I'm going to focus on the following 3 courses that I took, which were numerical analysis for multi-scale biology, supramolecular structure determination, and workshop in light and electron microscopy, which unfortunately is not provided in the same way because the professor actually passed away like shortly after I took the course, and believe it was 2014. But I believe that there is a course, now that I've looked at the new training material, there is a course that's sort of like it. I'm not necessarily sure if it's light and electron microscopy. But with that, the first course that I took was numerical analysis for multi-scale biology. It's the bioengineering, chem, math, and pharmacology cross-listed 276 course. And specifically, they taught us how to use mathematical tools to simulate biological processes in multiple scales. So throughout this presentation, you'll hear me talk about 2 of these tools repeatedly. And the first one is brown dye, which allows you to understand how small molecules and proteins interact, or proteins and proteins interact with each other. It's most specifically designed for small molecules and large proteins. And it gives you information about kinetics, specifically association rates, which tells you how quickly 2 molecules can come together and at what probability. And then I also used a tool called MCell, which if you've taken the course, you'll learn about this very, very powerful tool to simulate a cardiac system. And I'll tell you a little bit about that work as well. So the overall goal of the multi-scale biology course is to teach you to think in a multi-scale way. And the reason why it's called numerical analysis is because we use computational tools. And so there's an analogy that many people use called the computational microscope, which really allows you to use computers to peer into a chemical system to be able to examine that chemical system at multiple different scales. So if you have something on the scale of small molecules, proteins, you can use molecular dynamics. You're going to be thinking on the scale of femtoseconds all the way to microseconds. Well, mostly nanoseconds, but we can access microseconds to this present day. And if you're thinking about proteins working with each other, with them changing shape and being able to interact with each other or with small molecules, you're going to be thinking on the scale of milliseconds to seconds. And so the ability to think about this very multi-scale process is one of the advantages that I gained from this course. So as we know, this cellular structure can be broken up into both membranes and molecules. And through my work in that course, I was able to work directly with one of the professors in the interfaces training program, Dr. Andrew McCulloch, and Britton Boras, who was his student in bioengineering. And we actually published a paper called Bridging Scales Through Multi-Scale Modeling, a case study on protein kinase A. And this was my first ever paper. I was a co-first author. It was such an honor to be able to be part of this project. And if anybody doesn't know about protein kinase A, I'll tell you just a little bit about it, because I don't want to get too into the details about the protein system. I'll sort of give you an overview. Protein kinase A is made up of 4 proteins. It's a complex of 2 regulatory subunits, which are directly inhibiting a catalytic subunit, and their inhibition of the catalytic subunit, which is responsible for turning other proteins on and off through phosphorylation, is directly controlled by the binding of cyclic AMP. You can see cyclic AMP binds to the regulatory subunit. So there are 4 sites to which it binds, and overall 2 catalytic subunits are released. And so we can think about the problem of regulating pKa as a multi-scale problem. pKa is known to play a very vital role in cancers and a lot of different diseases, because if you control a protein that controls other proteins, you can influence disease. And so we sort of put together this this way of approaching this problem, and I want to highlight that we created this sort of pipeline of putting information together from molecular dynamics, talking about how proteins move intramolecularly, and getting information from that to be able to use in Brownian dynamic simulations, where we're talking about intermolecular simulations, how molecules move with respect to each other and how they associate with each other, and taking that information about their activation and moving it up the scale into kinetic protein Markov state models, and thinking even further up how we can take that information and port it up into both stochastic and deterministic signaling molecules of what's happening in the cell. So if we're thinking about individually cyclic AMP, how it interacts with protein kinase A, how protein kinase A interacts with other proteins in the cell, and how the cell function is then influenced, this is a very multi-scale process, and that was specifically shaped by this class. So one of the methods that we use in the lab of Romy and Morrow is molecular dynamics. And what we do in molecular dynamics is we break up the motions of every atom in the system into their bonding and non-bonding energetic potentials. And over time, what this gives us is the motion of very distinct pieces of the protein, which are called structural motifs, how they move relative to each other. And you can put all that information together through Markov modeling. And one of the tools that you'll learn about in the 276 class is brown dye. So what we use in brown dye to understand intermolecular associations, talking about small molecule like cyclic AMP associating with a protein, you use the chemical structure and you use the long range electrostatics, meaning how the molecule looks to another molecule with long range forces, what attractive forces are going to influence a molecule to associate or not. So what you're doing with Brownian dynamics is you are estimating the probability of association. So if you see here, that's my small molecule, that's cyclic AMP. This is protein kinase A. This molecule will either escape or it will associate to a specific region. And we'll talk a little bit more about that a little bit later. But that gives us kinetic rates, which can then be piped into larger whole cell models of how the R subunit and C subunit and pKa interfaced with each other. So that's one of the ways that my coursework sort of influenced this multi-scale perspective. We'll come back to that specific course and those specific tools a few more times. But I want to tell you about another course that I took, which is called the Supermolecular Structure Determination course. It's a bioengineering chemistry 283 cross-listed course. And it teaches you specifically about bioinformatics tools and mass spectrometry and how the structure-function relationship of proteins can be elucidated using these tools. And so I specifically used the knowledge that I gained about mass spec in my next project, which again was focused on pKa. So to give you a little bit more understanding, when we talked before about the way that the regulatory subunit is influenced by cyclic AMP, there is a structure out in the protein data bank of a protein. It's a small portion of the regulatory subunit. So this is your protein unit here, and this is cyclic AMP in the association site. I don't know, can you guys all see my cursor by chance? I'm not able to hear you all.
It doesn't look like it.
It
right?
doesn't.
We don't see it.
Oh,
You
yeah,
don't see it.
it's just very.
There it is, there it is. Okay.
Okay.
Found it. Yes.
Great. And so you see this small molecule, this cyclic AMP in here and cyclic AMP in here. So there is a structure of the regulatory subunit, and there's also a structure of the regulatory subunit in complex with the C subunit. And if you notice here, the C subunit on the right-hand side, which is R associated with C, there doesn't seem to be a way for the cyclic for the C subunit to be able to interact with the regulatory subunit R over here if cyclic AMP is around. So there's some sort of major structural change that happens. And for many years we had been sort of mystified by how the R subunit interacts with C in the entire sort of signaling cascade when cyclic AMP is around, because there is this sort of major structural reorganization. And so if we look back, a lot of people have investigated the system, and it's very apparent that if you cut off the A domain, which is happening in this top right figure, the small angle scattering, which is called SAX, is completely different. And what this tells you is sort of the shape of the molecule. This shouldering region disappearing in this top area definitely tells us that there's this sort of symmetrical, how do I say this sort of symmetrical property, right? It's sort of globular. But the shouldering region, when the B domain is present, which is the domain that's thought to regulate most of the function, there's this sort of opening and closing that happens, the shouldering region. And so if you mutate this B domain, you get the restoration of that sort of globular structure where you don't have the motion of that B domain sort of opening and closing. And so this work led directly to publication in the Journal of Biochemistry, where I examined the R subunit in that sort of holo conformation from that conformation that I got from the C subunit being present, basically deleted the C subunit. And then I ended up seeing the regulatory subunit kind of crack in half. It bends along this helix, and not only did it access that conformation, but that conformation was incredibly stable. And so this conformation happened very early on in the simulation, and it stayed there for the entire duration of the simulation. And not only was it able to stay for the entire time, but it gave us this sort of viable confirmation of how the C subunit can associate with the regulatory subunit and only the A domain, right? So there's a sort of like shouldering that was happening, which it was explained by the SAX data that was already out there. And so that specific class, that supramolecular structure determination class, allowed me to have an additional level of literacy with respect to hydrogen deuterium exchange. The reason that I discovered that this was important is because I went back to the HDX-MS papers and was able to see that in the C subunit, you have these unprotected areas. And those areas are completely unprotected if we are able to put the R subunit in contact with the C subunit. And so I was able to uncover with chemical simulation tools this confirmation that had mystified so many chemists for years. And specifically, this is the work where we say Anand et al. This is the work of Dr. Betsy Comabez, right, who had fought for this complex for so long, but the work was not really supported by the crystal structure. So this work specifically influenced the way that we understand how R and C subunit interface with each other. And it completely changed the way that we think about the B domain and what it's doing. And this has led to a significant amount of work. Thereafter, this work was kind of halted in 2007 because Susan Taylor and Betsy Comabez had two very differing opinions about what this structure looked like. And what actually ended up happening is that this work built a bridge again between both of the findings, which actually ended up both being correct. It's just that they were examining things from two opposite sides of the spectrum. So not only did we discover this hidden conformation, this flip back structure, but there are also implications for cyclic AMP association. So if anybody remembers, brown dye is a tool that allows you to see how a small molecule can associate to a certain domain. So that is cyclic AMP associating to the B domain. And there is this tool, brown dye, allows you to take the ligand and structure. You're taking the PDB structure and you're converting it into a charge and radius structure, where you're able to see the forces that cause the attraction between the two molecules. And the way that you do that is by getting this electrostatic description, which you get through the APBS software. And what happens eventually is that a molecule like cyclic AMP will associate to the specific domain, and it'll be called a reaction. So you have that complex is called an encounter complex. And what you need for that are the atoms in pairs, the distances between the two of them, which you can get from a crystal structure, or you can take your best guess. Or that molecule will actually end up escaping. And you're able to calculate the probability of whether or not it will react by having it start on that Q surface and seeing whether or not it will actually, from the B surface, to see whether or not it will actually escape from the Q surface. So overall, you're getting a reaction probability as well as a second order rate constant, and also a trajectory to see how often this molecule will associate and if it will or not. And so I use the crystal structure to describe the encounter complex. The crystal structure that had already been published back in, I believe it was 2007, if I'm not mistaken. And there were certain interactions that were repeated between the B and the A domain. So I kept those constant. I had an electropositive region of the interaction, which was specifically an arginine and the phosphate portion of cyclic AMP. I also put in a hydrophobic interaction. Electrostatics are not heavily influenced by hydrophobicity. They're more influenced by positive negative interactions and polar interactions, but I put it in because it was one of the things that I saw sort of being repeated between the A and the B domain. And then the last thing that I put in were the electronegative interactions between the glutamate. So I was able to take these, oh, I'm sorry, the year that it was published was actually 1995. It was based on the 1RGS crystal structure. My apologies for that. So overall, what we end up seeing is that when we did the electrostatics of these regions, we see, hope you guys can see this okay, right up here, there is a change in the electrostatics of that region where cyclic AMP binds. Notably, there's a much more electropositive region, right? The bluer binding pocket will show you that a negatively charged small molecule will more likely associate to that area. And the same thing sort of holds for the tetramer. If you're looking at R2C2, that same thing happens where the B domain flips backwards. And so when we were able to do BD simulations of this, we ended up seeing that the flip back structure actually neutralizes this preference. There's a pretty severe preference for the B domain if we're looking at between the two, and that flip back structure actually neutralizes it, which gives a path for cyclic AMP to bind to the A domain. And more specifically, if it's in the heterodimer, right, if there's just R and C, the flip back structure actually prefers the A domain association. It's much, much more accentuated. So with the discovery of this, we were able to sort of create a brand new method of activation for it and for PKA, and you have the flip back structure playing a role in the association of cyclic AMP to the A domain, which we didn't have any sort of idea before. And it's one of the intermediates that's thought to play a role specifically in that binding cascade. And so the last course that I took which unfortunately is no longer offered, is the light and electron microscopy course taught by Gina Sosinski. I'm not sure if Dr. Mark Ellisman is still teaching a course. I know that he's one of the professors listed in Interfaces still to this day, but he also was teaching the course with us. So what we learned in that course was about electron microscopy. It's a very valuable tool where you're taking a small sample. An electron beam is shot through that sample, and the small sample of what you're examining is sort of tilted in multiple directions. And what that gives you is a sort of three-dimensional reconstructed view of the system that you're studying. So the specific system that we decided to apply this to is the cardiac system. So the cardiac system is inherently multi-scale, right? You have this, as we discussed before, the heart is an entire organ that's made up of multiple levels of cardiac ultrastructure, specifically, the cardiac tissue, which is made up of cardiac myocytes, which are made up of myofibrils. And if you zoom down all the way to the cellular level, it's made up of a number of proteins in the system. And so what you're able to do is take some cardiac myocytes and get a reconstruction of them through I'm sorry, I think I skipped a slide. Yes, you're through electron microscopy. So that's exactly what was done back in 2009. Dr. Mark Ellisman with Dr. Masa Hososhima were able to get a 3D reconstruction of cardiac myocytes, and were able to reconstruct a three-dimensional view of that system that included mitochondria, it included sarcoplasmic reticulum. This is what it ended up looking like. So if you see here, you have T tubules, you have the mitochondria in pink, and you have the sarcoplasmic reticulum in yellow and green. And so if we look at that on the cellular ultrastructure level, what you see is that there's this sort of very, very regular pattern of interface of the T tubule, which is actually the plasma membrane, with the sarcoplasmic reticulum. And so those two are brought together at this region called the dyadic junction. And what ends up happening is that cell signals from the excuse me one second, let me just get some water. Cell signals from the extracellular matrix, specifically systems that are relaying the message that the heart should beat and the heart should activate, are coming through these T tubule structures, which are your plasma membrane, which are shown over here on the right panel in green. And they're physically coming into the cell and interfacing with the sarcoplasmic reticulum, which holds a bunch of calcium in there. And so that physical proximity, which I'll show you a little bit more about later, is something that's very, very important for the accurate firing of these systems to be able to activate at the right time. And so you have synchronicity of the heart beating and the lengthening and relaxing of your heart muscle systems. If there's any sort of disruption in that system, you end up getting heart disease. And so what we decided to do was to take this system and use MCell to understand it. If anybody has taken the Bioengineering 276 class, you'll know that MCell is a very, very powerful tool. It allows you to take all the molecules in a system that we saw before and to quantitatively investigate what's going on. And the way that you do that is by taking these sort of abstract concentrations and putting them into actual numbers, right? You have a certain concentration that will translate to to a physical number. So in the case of one of the major receptors in my system, ryanodine receptor, that translated to 96 receptors because we were able to see it in the EM images. You can take these diffusion coefficients, which are which will specifically influence how the molecules are walking through the system through a random walk with Brownian motion. You can confine them to a specific boundary and say that they are in this mitochondrion, they are in this sarcoplasmic reticulum, and they can only go in and out through interaction with other molecules. Reactions can be bimolecular, they can be unimolecular, reversible, they can be irreversible. And the models have to describe whether or not the molecule will react by itself, right? A protein can undergo a conformation without interacting with a calcium ion, for instance, or protein kinase A can open and close without interacting with cyclic AMP, or you can investigate whether or not those molecules interact with each other, how fast they will associate with each other. And that'll give you an understanding of what's happening in the entire cellular system. So what we ended up having to do was to take this very large system that had so many proteins in it and put in the missing pieces so that we can understand what's happening in the entire cardiac system. And what that involved was breaking down the system into the specific receptors. We had T-tubule fluxes where we had plasma membrane calcium ATPases that are pumping calcium in and out. We also had sodium calcium exchangers. Same thing there, they were pumping calcium into the system, and also voltage dependent calcium exchangers, which were the ones that were actually activating in response to an action potential. Opening the L-type calcium channels causes an influx of calcium, which will lead to the activation of that cardiac system. The other thing that we had to do was adapt a number of systems, specifically CIRCA. CIRCA is one of the systems that that clears the calcium and maintains the calcium level inside the cardiac myocyte. And we only had models that were treating calcium inexplicitly, right? Saying 2 calciums are taken. Well, MCell really needs things discretely modeled, 1 calcium at a time. So we had to do a bunch of adaptation there. And then one of the major proteins that we were looking at, the ryanodine receptor, which you see here on the right, had a number of reactions that described the opening and closing of this sort of fire hose system that causes calcium to flow in and out. And one of the advantages of using MCell is the ability to model all of these, you know, in this insane number of reactions that we're looking at here and discretely, right? Looking at every single calcium ion and what's happening in that system, the ability to model all of these things in a physical geometry. So the other thing that we did was we combined MCell with a with that geometry that I had shown you before, but we also made deformities. So over here on the right-hand side, if you see, we have a deformed T-tubule, a disruption of that dyadic junction, and we wanted to compare what happens when you have that deformation. And so what we ended up seeing is that chemical signaling is completely obliterated. This is the dyadic junction. You see these ryanodine receptors over here on the left and on the right. The open receptors are shown in white, and in the deformed T-tubule, you have the obliteration of activation. So on the left-hand side, you know, you have a number of ryanodine receptors that are activated next to each other, right? They're sort of neighboring ryanodine receptors are causing other ones to open and causing the system to be able to communicate properly. And what you ended up seeing in the system when there was a deformation is that that doesn't happen as often. So it gave us a understanding of this very complex system that sort of came down to whether or not the cardiac ultrastructure was conserved. The other thing that we ended up having as a result of the system is a completely new way to visualize the cardiac system. So not only were we able to see the beautiful images that Hayashi was able to take out, but we were able to also understand the biological context and the structural context of these small molecules. So these are my L-type calcium channels on the plasma membrane on my T-tubule. This is my my ER, my sarcoplasmic reticulum, and the ryanodine receptor proteins in there. And what we were able to do is to unite a number of tools, specifically CellPack, and Molstar was actually able to be used to visualize the system. So the overall message that I want to tell you is that if you utilize this program, you will be able to be very, very successful in learning a number of tools, right? I just described 4 or 5 different tools to you that I was able to do in my short graduate career. And owing to those 3 classes, I was able to be very productive in my graduate career to understand a number of very, very different topics in very different spatiotemporal fields, and illuminate missing information, specifically using the tools that were taught to me in the bioengineering classes and in the chemistry classes that I took. Those classes, you can take courses throughout your graduate career and sort of call it a day and just, never use it again. But I was really set on using that information to be applied to the research problems that I was studying. And I was very fortunate to be able to have really great mentors that were very encouraging. Some of these problems were very, very hard to solve, and they were very, very supportive. So it was a unique opportunity to be able to investigate something that nobody else sort of had the time and space for. But because Interfaces not only funded me, but gave me the tools to be able to do these investigations, I was able to make a very significant impact in a number of different fields. And I have more work that I haven't shown here today, but these are 2 or 3 projects that were specifically influenced by the training program. And so if you leverage these tools, you will be successful. I guarantee you that. I'm a living example of how that happened. And in going into the second portion of my talk, which will conclude shortly, I'll tell you a little bit more about how the training program actually influenced me to develop professionally, both in graduate school and outside of graduate school. So part of the Interfaces program is the funding, right? The reason why the Interfaces program has been so successful is because of students like me that came out that were able to make impactful scientific findings. And that paper, every single paper that came out of my training is sort of a receipt to a funding agency to say, hey, renew our training program. So I was fortunately able to help with that. And my professor had me, write her portions of the training grant and said, give me a summary of how your work was directly influenced. And so this presentation so far has been, evidence of that. But I was able to learn how to write grants and learned how to fund my research, right? Because every single time I had to renew my funding with Interfaces, I had to sort of write a summary that said, this is how the coursework has directly influenced my work. But it also taught me the very valuable skill of being able to write grants. After graduate school, I worked in the capacity of writing grants with other scientists. So the ability to talk to different scientists, right? I was working at one point with somebody in mass spec, and then I was working with crystallographers, and the ability to communicate with those types of different audiences and, computationalists, they all use different languages. So being able to think multidisciplinarily and being able to communicate with a wide range of scientists is definitely something that that resulted from my time at the Interfaces training program. And then the other really cool part of being part of Interfaces was being able to teach the courses that I took, right? There were times where I was brought in as a guest lecturer and I was able to talk about my work. I created whole assignments based on based on the flip back structure that I had just taught you about. I was able to make assignments for classes where students were able to use the same tools that I used in my research, for them to be able to replicate that work for themselves, for them to be able to visually see this with, with VMD, which is one of the tools that you use to visualize some of these systems. So it was really awesome to be able to talk about my work and give back to the program that gave so much to me, right, by training me. And I was able as well to be a mentor to many graduate students. I mean, students that were in a master's program, students that were in PhD programs. There are even students in my old lab at the Amaro lab that are part of Interfaces to this day, and they'll call me and they'll say, how do I sort of put these tools together? How do I use these tools in my research? And it's so valuable to be able to draw from my own experience and to tell them, hey, have you thought about doing this before? Have you thought about doing that before? So being able to mentor other master's, PhD students was probably the biggest gift of Interfaces, being able to sit in that mentorship role, because if you have a good mentor, you know what it's like to have that very wonderful relationship, and being able to step into the space of being a mentor was very, very valuable for me. And so after I graduated, I worked in a number of spaces, but present day, I actually work at the US Patent and Trademark Office. And at the USPTO, what I have to do daily, I'm a chemical patent examiner. What I do daily is I read the most cutting edge technology about, whether it's small molecules or different types of chemical compositions that are to be used to solve problems in everyday life. I have to understand that chemistry. And what that requires is this ability that Interfaces gave me to think about vastly different concepts, and to have this interdisciplinary mindset to be able to think about these problems. And as a chemical patent examiner, I use a number of advanced tools to do my structure searches. And because I was constantly developing materials for students and for my mentees to use, I'm also applying that skill of developing those same types of materials and helping my colleagues who are doing the same work as I am. I'm a new examiner, and I've already led 2 classes where I teach people how to use these advanced tools. And that's a direct consequence of taking the courses that I did, right? Where I learned how to use vastly different tools, learned how to apply them to my work. So being able to develop that kind of material for other patent examiners and being able to teach them how to use it, I have this literacy for coding that many people do not have coming out of graduate school. And that is 100% influenced by the courses that I took at Interfaces and the research work that I ended up doing in my graduate career. So it's given me a leg up. I've been very, very successful as a patent examiner thus far. And so all of this stuff is, just to conclude overall, all this stuff that I've spoken to you about today is directly influenced by the Interfaces graduate training program. The coursework and experiences that I had, I worked with physicists, I worked with neurobiologists, I worked with computer programmers, I work with people that work in the graphics and visualization space. Being able to communicate with that many people and to be able to collaborate with that many people made me a very successful graduate student and has led me to be able to communicate with a vast audience of scientists, of students. And had I not been in interfaces, I would not have been able to do that, hands down. Prior to coming to graduate school, I worked in hospitals, and that required a completely different vocabulary and a completely different mindset than computational chemistry required. And interfaces really set me up to be able to communicate with a large number of people about my work and about the importance of studying these different topics that I was able to work on. Additionally, my experiences in teaching in interfaces and mentoring other students and being a mentee was invaluable, because the feeling that you get when you give back to younger scientists, because any scientist will tell you, we have a time limit on us. The most important thing that we can do as scientists is communicate our work and teach the younger generation, teach younger graduate students how to think about problems critically and how to break things down and how to solve these problems. And the ability to fund your research, right? To convince funding agencies that your work is worthwhile, because research doesn't get done unless it gets paid for. So those two very, very invaluable experiences have influenced my success as a scientist throughout my life. And had it not been for interfaces, I don't think I would have been as successful as I am. Well, I know for a fact that I wouldn't have been as successful as I am, because it really made me a leader. It made me into the type of scientist that takes initiative, that says, hey, this is a problem and I'd like to solve it. And even in my training academy for the USPTO, I was a person who took charge and said, hey, if there's a problem, this is how we're going to solve it, specifically with the chemical tools that I'm using in my work. So with that, I thank you all for inviting me to give this lecture. I hope I used my time well. I'd like to thank you all for your attention, and again, thank you all for inviting me here.
Thank you. Thank you very much for that. Who's got questions or comments or anything? Some of you have taken these courses already, especially 276. That seems to be one that everybody takes a lot. What kind of questions or comments do you have?
About chemistry, so I shouldn't be asking.
I started to say, Katie's always good for a discussion.
Yeah.
We can always count on her. This position is new for you, is that correct, Dr. Hirakis?
Yeah, it's a new position. I'm not sure if I'll be able to keep it, because with the change in administration, they're causing a lot of people to return to in-person work. And unfortunately, I'm affected by a permanent disability. Yeah, so it's going to be difficult for me to relocate down to Alexandria. So I'm not sure how long I'll be at the USPTO, but it's been great so far. I love my job. It's awesome. So anyways, yeah, I'd love to take your questions. I'd love to hear your guys' experience. I kept the presentation short because I do want to hear from you all. I want to hear how you guys are doing in interfaces, whether or not you like the coursework, have you been applying it to your work, etc.
We do have some people here who are in their second year on the training grant, and some who just started in the fall. So we've got a little bit of a mix here today. Does anybody want to offer up what their experience has been like? We're getting ready for the annual RPPR right now, so I'm bothering everybody for their progress reports for the NIH, and speaking of funding possibly being removed, we just got this renewed, the interfaces, well, not the interfaces, the NIVIB training grant that we receive our funding on just got renewed last year with a perfect impact score of 10.
Congratulations.
I couldn't, Dr. McCullough was so excited. We were all so excited. And so, and that is a direct reflection of people like you, that you are the reason that this we got that score. So, you know, people take this training, they participate in this program, and look what they have been able to accomplish. And yes, we're gonna give you some more money, so yay. So I'm glad we got that done last year. And, so that's what we want to try to be able to convey to the students, is that you can experience this as well.
Yeah.
And,
By the way, I just came out with a new manuscript, so if you guys need to add that to your
report for the NIH.
yeah, I'm happy to send it to you.
Yay, that would be fabulous. Thank
I
you
think
so
you guys are
much.
typically listed in the acknowledgements, I'm pretty sure.
Okay, excellent. Thank you so much for that. Anybody?
Sure.
So I'm in my second year of the training grant. I'm in the bioengineering department. And it was super cool hearing how you were actually able to use what you did in class for multiple publications. That was really nice. But I was wondering kind of how, it was cool to hear how at the patent office, your knowledge was really valued and it was necessary to have people with that knowledge. But how did you make that transition from being in a research lab to reviewing other people's patents? It seems kind of like a change of scenery.
Totally, absolutely, yeah. So, you know, working at the USPTO, there was a patent training academy. So we were specifically trained on what it means to read a patent application, to examine something. Unfortunately, we didn't have specific training on what's called our technology center, right? Our art unit. So in patent speak, art is something that is used to design the chemical species that you're describing in your patent application. And so what my responsibility is to examine that application, go out and determine whether or not there is any artwork, any prior art that sort of is very similar to the present application. And so that is a skill that requires using tools that require you to use programming languages, right? I didn't know these programming languages coming in, but because I was able to learn so many different tools in the 276 class, I had this ability to quickly learn a tool and quickly learn how to use it. And so within the first 2 weeks of doing this, I wrote up scripts and I created tutorials that my coworkers use. So it was really great to be able to use those similar types of skills and to use them in my work. So it is a vastly different space of operation, but the skills are the same, right? Being able to use a new computer program to apply it to a different problem and solve that problem are the same. Right? They're just sort of in a different mask, if you will. But yeah, I hope I answered your question. It's definitely very different in research, right? You don't have the one thing about patent examination is that we have fast turnover times, right? You're looking at an application, you have to read it and you have to get it out the door and decide whether or not you're going to reject or accept, and to grant somebody a patent, right? And so the one thing that I miss about research is that luxury of time to really sit and read and learn. And oh gosh, I would love to do that for every single piece of technology that I'm examining and every invention that I'm examining, because all of them are really cool, right? Nobody would be paying all that money to apply for a patent if it wasn't cool technology. So definitely leverage that luxury of time that you have right now to be able to sit comfortably and learn everything that you can about the system that you're working with. It's one of the things that I miss about graduate school. My PI used to tell me all the time how much she missed graduate school. I was like, this lady's crazy, but she's right. She was totally right. Yeah, so I hope I answered your question. Let me know if you have a follow-up.
Yeah, no, it's interesting, like you said about having to have the feedback so quickly. Because Cause my lab has submitted some patents, so I've seen it from that side of things, but then also from the other side where you have all of these things coming in. And like hearing about all the CRISPR patent dispute and like there's just so many interesting things going on there.
That's the reason why our jobs at the Patent and Trademark Office are really important, right? Because if there is any sort of intervening art, right, somebody else did this beforehand, there's going to be a nasty, long litigation, downfall as a result of that. So it's really important that we do our due diligence when we're examining the patent applications initially. Because if we grant somebody something and then somebody comes in and says, "Hey, you're infringing on my work," it can be gnarly. So, are you in the chemistry department?
No, I'm in bioengineering. My lab's in pharmacology and chem biochem though.
Okay, cool, got it. Who's your professor?
Joe Schoenberg, he's fairly new, so I don't know if you.
I have heard of him. I visited
Okay.
campus recently. Yeah.
Oh, nice, okay.
Any other questions?
Shit, yeah. Hi, I'm Katie. I'm in the bioengineering department. I work on imaging, but like the whole body imaging, so, and so not on the same scale. But I guess we haven't taken the numerical analysis class yet as next spring, I think. But I was just wondering, you talked a lot about the tools, and all these tools have all the like numerical analysis, the modeling, like mathematical side of it in the background, but do we get to learn a lot about like the actual theory behind that more so, or is it just learning all the tools? Because I
So
can imagine there's so many tools in so many different areas.
yeah. So you will have at least one lecture on the tool itself, if not two lectures on the tool itself and the theory behind it. And then you will learn hands-on in the lab how to use that tool.
That's cool. Is it all like like that scale of things? I don't know.
Yeah,
I don't know that much about this stuff, so it's a little hard to relate.
You'll get to the whole organ scale if I'm not mistaken. I don't know the current curriculum. You'll get to the whole organ scale too. But you'll learn how to use some of the tools that I taught you about. And if you interface with Tom Bartol, who teaches MCell, tell him I said hi and tell him that you had a lecture on the tool. He was also co-author on the recent paper that I showed you guys about the cardiac system.
Awesome. Thanks.
Yep. Yeah, thank
I
you.
took 276 last year, so it sounds like it hasn't changed that much since you took it.
So there's a lab component too? And
I mean, it's
like
it's,
a hands-on.
yeah, there's a hands-on, and there's a project at the end where you have to combine multiple tools.
Yeah. So actually this work came out directly from that class, right? This was sort of, the project that I whipped up in my second year when I was in interfaces and put it together and said, this is what I want to do for this project. And it ended up being a major part of my thesis. It ended up being a massive manuscript that took 12 years to publish. And so I want to stress to you that you can work these classes into your thesis, right? Then they're not just classes that you take to get a PhD specialization. And actually, I'm looking right at my degree. My degree says my PhD in chemistry, and it says specialization in multi-scale biology. It's a special degree. Like if you graduate from the chemistry department, chemistry and biochemistry, you'll have a doctorate in chemistry, right? But this will be a specialization in multi-scale biology. It's an additional set of coursework. It's an additional set of requirements that are set out for you to be able to have that hat and wear that hat and say, my work is specialized in multi-scale bio. So it's definitely a step up. I want you to know that you guys are definitely privileged to be part of this program. And I loved it. I hope that you all have a very, very fruitful experience with it as well.
Thank you very much,
Thank
Dr. Hirakis. We got to wrap it up. Thank you so much for your time.
Thank
And
you.
please let us know if we can return the favor in any way.
Well, if anybody would like to get in contact with me, I'm happy to stay in contact with you all, especially with the new trainees. If you'd ever like to have, email correspondence, you're more than welcome to pass my information to everybody.
Thank you so much. I'm trying to get an alumni network up and running. And so we will definitely be contacting you and asking you to join that group officially. Thank you so much for your time.
Yes, thank you very much.
Have a great weekend. Thank you.
Thank you. Bye-bye.
Bye-bye.